According to the World Health Organization, an estimated 45 million people are blind worldwide. A common cause of blindness is loss of photoreceptors or light-sensitive cells of the retina. The retina is the layer of tissue at the back of the eye that converts images to nerve signals and sends them via the optic nerve to the brain. When the light-sensitive cells of the retina, called rods and cones, are damaged by disease, as shown on the right, the brain no longer receives signals from them. But the retina also contains other cells that do not themselves respond to light, but which process signals from the light-sensitive photoreceptors before they are transmitted to the brain. These cells can survive even when the photoreceptors are destroyed. So what if you could make these surviving cells sensitive to light? Could you restore vision? Alan Horsager, a neuroscientist at the University of Southern California, and Ed Boyden, an investigator at the McGovern Institute for Brain Research at MIT, set out to answer this question with a new tool called optogenetics, pioneered by MIT researchers. In optogenetics, researchers take a piece of DNA that encodes for a light-sensitive protein. In this case, the DNA was taken from algae that naturally sense and swim towards light. Then, DNA is placed into neurons using a safe and effective method for delivering genes, the same kind being used in human gene therapy trials. The neurons express the DNA, manufacturing the light-sensitive protein molecules and installing them in their surface membranes. Like tiny solar panels, these proteins respond to light by generating electrical currents. This causes the illuminated neurons to produce electrical impulses that mimic the brain's natural signals and can be transmitted to other neighboring neurons. In this study, researchers used optogenetics to find out if they could restore vision in blind mice. To do this, they tested a mouse's ability to swim out of a chamber in which the escape route was indicated by light. Blind mice, as shown here, cannot follow the light, so they must circle around the chamber exploring for an escape route by touch alone. Mice treated with optogenetics, however, learned to escape the chamber as fast as mice with normal vision, and they were on average nearly 3 times faster in escaping the chamber than untreated mice. In addition, the researchers found that the blind mice expressed the light-sensitive gene for at least 10 months, the duration of the study. That's important for human therapy since retinal blindness is a chronic condition. They also found no evidence of harmful side effects, which is an important step towards human clinical trials. EOS Neuroscience, a company founded by the study's co-authors, hopes to begin testing this tool in humans with retinitis pigmentosa within 2 to 3 years. They also believe that this therapy could have a dramatic impact on patients with age-related macular degeneration and other retinal disorders.